Disclaimer: this content is created to assist students with their studies. It is not affiliated with or endorsed by Cambridge. Workbook and learner's book questions are used under the assumption of fair use for educational purposes.
Chapter 1: Photosynthesis and the Carbon Cycle. Here we go with exercise 1.1a, how light level affects photosynthesis. Arun wants to find out if plants photosynthesize more quickly when they are given more light. In this activity, we need to think about fair testing. A fair test means we only change one thing and keep all the other things the same. So what will Arun change? The light level. What will Arun measure? The number of bubbles made by the plant. And what must stay the same? The plant type, the water, the temperature, and the time of the experiment. That's how Arun makes sure his test is fair. Now, look closely at the diagram. We can see three setups: apparatus A, apparatus B, apparatus C. Each one has the same water plant in the same beaker of water with a tube at the top to collect bubbles. And those bubbles? They're oxygen, made during photosynthesis. The more bubbles we see, the faster photosynthesis is happening. Now let's see what Arun does with his three setups. He puts apparatus A right next to a window, so it gets bright light. He puts apparatus B in a shady corner of the same room, so it gets less light. And he puts apparatus C in a dark cupboard, so it gets no light at all. By changing the light levels, Arun can compare how many bubbles each plant produces. More bubbles means faster photosynthesis. Fewer bubbles means slower photosynthesis. Now let's check what Arun discovered. First, remember fair testing. Arun kept the type of plant the same, he kept the mass of the plant the same, and he kept the temperature the same. That way, only the light level was different. He left his three setups for 2 days, and then measured the gas in each tube. Here are the results. Test tube A with high light collected 18.3 cubic centimeters of gas. Test tube B with low light collected 7.2 cubic centimeters of gas. Test tube C with no light collected only 0.5 cubic centimeters of gas. Wow, big difference, right? So what conclusion can Arun make? Plants photosynthesize faster when they have more light. Now we move to exercise 1.1b, the effect of different colors of light on the rate of photosynthesis. Marcus wanted to investigate which color of light helps a plant to photosynthesize the quickest. Here's how he set it up. He placed water plants in test tubes and shone a lamp on each one. To change the color of the light, he covered the lamp with different colored cellophane sheets. Then he counted the number of bubbles released in 1 minute. And to make it fair, he repeated the test 3 times for each color. Here are his results. Red light cellophane, 10, 12, 11 bubbles. Green light cellophane, 4, 5, 6 bubbles. Blue light cellophane, 8, 12, 10 bubbles. Colorless, no cellophane, 11, 13, 12 bubbles. So now we can compare. Which color of light made the plant photosynthesize the fastest? Now let's go through the questions about Marcus's experiment. Question 1. What was the variable that Marcus changed in his experiment? The answer is the color of the light, using different cellophane sheets. Question 2. What was the variable that Marcus measured in his experiment? The answer is the number of bubbles produced in 1 minute. Question 3. List 3 variables that Marcus should have kept the same in his experiment. The answers are the light intensity or strength, the amount of water plant, and the temperature. These controls made sure the test was fair. Question 4. Here are Marcus's results. In white light, the plant made 11 bubbles, then 13, then 12. That's a mean of 12 bubbles per minute. In red light, the plant made 10, then 12, then 11. That's a mean of 11 bubbles per minute. In green light, the plant only made 4, then 5, then 6. That's a mean of just 5 bubbles per minute. And in blue light, the plant made 8, then 12, then 10. That's a mean of 10 bubbles per minute. So overall, photosynthesis was fastest in white light. It was nearly as good in red light, slower in blue light, and the slowest of all in green light. Now Marcus takes his results and draws a bar chart. Look at the colors of light. Red gave around 11 bubbles per minute. Green gave only 5. Blue gave about 10. And white gave the most, 12 bubbles per minute. So what conclusion can Marcus make? Plants photosynthesize fastest in white light. This shows us that plants need the right kind of light to make food efficiently. Now it's time for a challenge. Exercise 1.1c, turning an idea into a hypothesis that can be tested. Here's the idea. Carbon dioxide is an essential raw material for photosynthesis. So if we supply extra carbon dioxide by bubbling the gas into the water, maybe the plant can photosynthesize faster. So how do we turn that idea into a testable hypothesis? Simple. Water plants give off more bubbles per minute when they are given more carbon dioxide. That's a hypothesis. It's clear, it's testable, and we can design an experiment to check if it's true. Let's set up the variables. The independent variable, what we change, is the amount of carbon dioxide. The dependent variable, what we measure, is the number of bubbles per minute. And the things we must keep the same, the controls, are the light, the type of plant, the temperature, the amount of water, and the time of each test. By keeping all these the same, we make sure our experiment is fair. Next, we'll draw a labeled diagram of the setup, make a results chart with headings, and write down a prediction. So what do you predict? More carbon dioxide should mean more bubbles, and that means faster photosynthesis. Here's the method Arun could use. Place a water plant in a beaker under a funnel with a test tube to collect bubbles. First test with no extra carbon dioxide and count bubbles for 1 minute. Then add a small amount of carbon dioxide and count bubbles again. Then add a larger amount of carbon dioxide and count bubbles again. Repeat each test 3 times and record the results in a table. Work out the mean for each CO2 level and compare the results. Now let's check the results. With no carbon dioxide, the mean was 5 bubbles per minute. With a low amount of carbon dioxide, the mean was 11 bubbles per minute. With a high amount of carbon dioxide, the mean was 18 bubbles per minute. So what's the prediction and conclusion? The more carbon dioxide we give the plant, the more bubbles it will make. And that means the plant photosynthesizes faster. That's the end of exercise 1.1c. For more Cambridge book exercises, visit Patreon/Cambridge Answers. See you in exercise 1.2.